under advisor Dr. Vasantika Singh, thanks to STEM Over these past 6 weeks, we learned about RNA sequencing, how to analyze gene expression data, normalize large data sets, identify surface markers, and much more. To apply our learnings to the real world, in a project-based application, we decided to analyze the application of the promising CAR T-cell therapy on pancreatic cancer. Chimeric antigen receptor, one second, let me switch the slide. Yeah. Chimeric antigen receptor, or CAR T-cell therapy, is a type of cancer treatment that uses human T cells with engineered receptors to precisely target specific antigens found in cancer cells. This is done so that the cancer may be targeted by the patient's own immune cells. This is known to be a promising immunotherapy for blood cancer types, but efficacy in other cancer types is limited by off-tumor expression of target antigens. Why is CAR T-cell therapy needed? Classic chemotherapy indiscriminately harms all cells. This means that healthy cells are also harmed, which can weaken patients further. Classic chemotherapy is not always effective as well, so alternate methods are needed in these cases. So why is precision targeting needed? Precision targeting is needed in CAR T-cell therapy to prevent healthy cells from getting attacked if they possess the target antigen. For example, if an antigen present in pancreatic cancer is also found in the lungs, damage may be caused if the T cells target the cells in the lung as well. So current limitations of antigen escapes. So the problem is tumors can stop expressing the target antigen or mutate to avoid detection by CAR T cells. Logic gated CAR T cells. We specifically opted to use the AND logic gate in which both of the antigens chosen must be expressed in order to be targeted by the T cell. Let me switch the slide. We identified pancreatic adenocarcinoma as our target cancer due to the treatment difficulties which make it the cancer with the highest mortality rate. The survival rate is only 13% according to the American Cancer Society. In fact, pancreatic cancer is the third leading cause of cancer-related deaths in the United States. Pancreatic cancer, luckily, also has a high number of data sets available in, well, easy access to the public. In order to identify surface markers for the T cell logic gate, we analyzed bulk RNA-seq data from databases like TCGA and GTEx, normalized expression across tumor and normal cohorts, and filtered for protein coding membrane localized genes, using other databases like UniProt and the Human Protein Atlas. Expression analysis applied stringent thresholds, high expression in tumors and low to no expression, less than or equal to 5 against protein level evidence for surface expression from the human surfaceome. Expression profile was translational relevance. The most promising antigen pair we found was CCAM5 and GPR119. Tricia, would you like to talk about our results?
Yeah. So CCAM5, which is carcinoembryonic antigen-related cell adhesion molecule 5, is a cell surface glycoprotein involved in cell adhesion. CCAM5 has a very high expression in pancreatic cancer while sparing the normal pancreatic tissue, which is data based from TCGA. It is low to absent in most normal adult tissues except for the gastrointestinal tract epithelium at low levels. And due to this, the tissue that needs to be protected is mainly the gastrointestinal tract. CCAM is a good target due to its low to no heart and nerve tissue. The acute toxicities in the gut, however, prove highly detrimental in treating patients. GPR119. as GPR119 has a medium to high expression in pancreatic cancer and has no expression in the gastrointestinal tract. Therefore, we can use a antigen pair of CCAM5 and GPR119 in an AND-gated CAR T therapy to precisely target pancreatic tumors. So, we really enjoyed participating in the Summer 2025 Horizons Quest program as it was both challenging and rewarding as an experience. Working on the problem of designing safer CAR T therapies pushed us to apply computational skills to meaningful biological questions. We learned how to integrate large-scale data sets, as well as filter and analyze the gene expression and translate those results into therapeutic design concepts. This project gave us valuable exposure to the intersection of cancer biology, bioinformatics, and immunotherapy, which were areas we didn't have much knowledge in before. What we valued the most throughout the mentorship was the mentorship we really received. Collaborating with an advisor, Ms. Vasanthika, was amazing as it provided structure and guidance, and it also helped encourage our own independence and problem solving. We learned not just how to conduct analysis, but we also learned how to think critically about data quality, safety, and relevance. Overall, this internship strengthened our abilities, deepened our We really want to thank Dr. Vasanthika Singh and STEM peers for the opportunity they provided us.
Great job. Yeah, wonderful. Yeah, wow. Do you guys know what was the age of these patients that you guys selected for the vulcanism? Like age or genders?
Yeah, we just took like normalized data off of like HPA, TCGA, and GTEx. So I think it's like it covers like the majority of like all ages that normally have
All,
pancreatic cancer.
okay, all ages. So yeah, maybe the next, if you guys have like maybe the age bar like this is 20 to 40 or 40 to 60, maybe which is going like more or like less for this disease. So anyone have a question for them? I think so, no, but you guys did very wonderful job. And in my hospital where I work, there is really a lot of clinical trials going on the basis of the precision CAR T cell therapy. This is one of the most famous ongoing immunotherapy program that's going to be that's right now is running in Children's Hospital of Philadelphia. It's not especially for this disease, but there are like a lot more disease where like the team are working and they are developing the targets for the antigen. And the CAR T therapy is one of the most rewarded therapy like they wanted to do and like do the in the patient and see like how these cells are going to be response against those antigens. Yeah, awesome. And thanks to your mentor, like they also like, and your team, they for this like wonderful of the presentation and all the work you guys have done. Thank you so much. So with this, I think we have all presentation done. So everybody, thank you so much and for your time. And I would really appreciate like your effort if you go back to your the main room where we are going to be have like together and then we can ask like chat about each other. Yeah. So please feel free to go to and join into the our main room. And thank you so much.